Lovely. Thank you very much. Thank you for inviting me, Munir and Genomics England. I'm going to talk to you about three things. I'm going to give some background on human knockouts and some basics. What's a knockout? How many do we expect to see? That sort of thing. Then I'm going to give one specific industry example of where we've recalled a person with a knockout who's been invaluable for drug development. And then the last bit, I'm going to talk to you about the Genes and Health project and that in general and to do with knockouts. One thing I want to make right from the start is we've got two ends of the spectrum. So Genomics England has been studying rare disease mostly, and so often those are children with rare disease, and the prior probability of the knockout that you observe causing the disease is very high. We have been studying adults who have been ascertained in the community, so have population prevalence of disease, but probably won't have rare diseases. And if you see a knockout, the prior probability of that being associated with a disease phenotype must be 1,000 times less than it is for the rare disease collected cases of Genomics England. So there are absolute ends of the spectrum. So we see knockouts that don't seem to have any disease phenotype. And just to get that concept over. So we're going to do a bit of a triple act today. I'm giving the talk now. I'm going to give rather an overview. Dr. Hilary Martin is going to talk after lunch, I think, and do some of the more fine details as to how many people you might need to sequence to see so many knockouts and some population characteristics. And my colleague Richard Trembath, who's a co-lead on the Genes and Health study, is also around for conversations and discussions. First bit of the talk, what's a knockout? Daniel MacArthur published a paper in Science in 2012 showing that we all have loss of function variants. So I have loss of function variants. We all have about 100 loss of function variants, mostly in the heterozygous state. If they're common frequency invariants, we can have a complete knockout, i.e. both alleles are knocked out. But in outbred people, these are mostly in smell and taste genes, and this is why we can't smell what a dog can smell, for example, because we're losing these through genetics. But if people's parents are related or consanguineous, we know that you can see two copies of a rare variant, and I'll talk to you about this. And when we're talking about knockouts, we're talking about loss of function variants, and in particular, I mean stop codons, frameshift indels, and things very close to a splice site. There may well be knockouts in the promoter region or in other bits of the genome, but we're hopeless at annotating them at the moment. And obviously there's many decades of rare disease research into human knockouts. There's much less research into healthier adults, which I will show you. And so these are the type of variants where, when I say knockout or loss of function, I'm going to mean predicted from the genome annotation loss of function. We may not have proved it, and I'm meaning just these types. There are other types of knockout, large gene deletions, promoter and other bits of the genome, which I won't consider. The first big study was 2015. Actually, I didn't. This is on the way to the airport, and I went to see the Viking ship and to look at the climate change effect on all the volcanic rocks on the way to the airport in Iceland. I don't know if you've done that, but I missed out on the blue pool. But anyway, in Iceland, nice paper showing that in essentially the entire Icelandic population, which is a bottlenecked population, so at some point the population came down to a very small size and then increased again, so enriched for specific variants. They found 1,000 genes with knockouts, and this required the most enormous sample size. And then we published in Science in 2016, and you've already been shown the other paper in Nature 2017, sequencing adult humans at greater scale, but using consanguineous populations, which is a much more efficient method to find knockouts. And actually, we're both now working together, and there was a nice Nature Reviews Drug Discovery editorial on tapping the goldmine of human genetic knockouts, which I guess is one of the reasons for this meeting. So in our study, we sequenced, exome sequenced 3,000 people who had mostly stated that their parents were related from Bradford and Birmingham. The Bradford individuals were mothers ascertained in the antenatal clinic, so they were pregnant, and clearly that means they're a healthier population. And we found 1,000 knockouts. So I'm going to be just talking about the rare variants. I'm not really interested in smell and taste receptors, which are the common variants. It's the rare variants that are of interest. And we found about 1,000 knockouts in 700 genes and 800 individuals. We found one person with 7 knockouts, and we used a simple transcript annotation, GenCode basic. There's a whole load of transcripts in the genome databases, and there's a question as which you use. In contrast, and actually somewhat similar results, this was a study of unselected adults, 10,000 from Pakistan, case and controls for myocardial infarction, and actually found pretty similar numbers of knockouts when you contrast for the differences between the study. And the reason why they were 18 months behind publishing, even though we were both out on bioRxiv in the same week, is that Nature made them do manual annotation review of 13, 17 genes, which I'm very grateful to them for doing that, and pleased that we didn't have to do that. And some of the key findings from the study, we reported a knockout in a gene called PRDM9, which was thought to be critical for human meiotic recombination hotspots. And actually we found a mother who lacked this gene and was healthy with 3 kids. And actually the recombination in the genome just happened at different places. We did a little study on drug target indication pairs and showed that drugs where you have a knock, this is building on previous work from GSK and others, showed that if you had a knockout in a drug target, this was more likely to be approved from phase 1 to final approval than genes without knockouts. This is a safety indication if an adult human exists with a knockout, and it gives you a suggestion that the drug might also be safe. We did some looking at electronic health records of primary care and showed that there was no difference in prescription rate or doctor consultation rate. These are some of the best easily available measures of general health, comparing people with knockouts to people without. You'll remember that we selected people from the antenatal clinic, so this is not the same as a population with rare disease, and this would look very different if it was a population with rare disease, I expect. And then we looked at 54 genes with Mendelian, which we thought were actually Mendelian recessive genes. These are obviously going to be very enriched for errors, and we found that 16 of the errors were genome annotation errors, exon intron boundaries in the wrong place, things like that. There weren't any sequencing call errors. It was all genome annotation. But actually only 9 of these individuals had anything in the electronic health record that was compatible with the supposed monogenic disease phenotype. And this partly reflects incorrect data in OMIM, people reporting monogenic disease, whereas actually these days the evidence would not be considered adequate. The PROMISE study found, they showed the example of an ApoC3 knockout, which is involved in triglyceride metabolism, and this is a human study, 6 people with knockouts versus 7 people without, and if you feed people a fat diet, your fats in the blood go up, and if you have ApoC3 they don't. And then there were some predictions on how many people you needed to sequence to find 10,000 genes with knockouts, and actually Hilary is going to talk about this afternoon, so I won't do that. And obviously there's a lot of interest in human knockouts in drug development, not least the enormous effort that Regeneron is putting into this, and this is a finding in New England Journal. There's a number of other examples like this as to knockouts giving insights into new drug development here for cardiovascular disease. And can we use genetic data to improve drug development, and in particular the huge cost of a failed late-stage clinical trial? So I'm going to give you one example of one individual with one knockout, who we've recalled, which illuminates some of this. This is some work we've done with Alnylam Pharmaceuticals, who are interested in a disease called primary hyperoxaluria, which is a genetic disease of children which gives renal failure due to calcium oxalate crystals forming in the kidneys. And the only current treatment is a dual liver-kidney transplant, which obviously comes with a lot of difficulties and morbidity. And Alnylam have developed Alnylam have developed a drug called lumasiran, which is a RNAi therapeutic, which targets another. I'm not going to go into the. You're probably pleased. I'm not going to go into the detailed biochemical pathways, but it's a downstream enzyme in the glycolate pathway, and it's this oxalate that forms the crystals in the urine, and this goes on in the liver. And lumasiran targets glycolate and knocks down glycolate. Tracy McGregor, who's a clinical geneticist from Alnylam Pharmaceuticals, looked at the list of knockouts from our Science 2016 paper and said, oh, you've got a knockout for HAO1, which is this gene glycolate oxidase. Please, can you tell us more? And their particular questions were about the lumasiran. This is something that's going to need to be given long-term to people with primary hyperoxaluria. It's going to need to be given for life. And actually, is it safe to knock down this glycolate oxidase HAO1 gene in humans? And are there any other effects of the HAO1 knockdown? And actually, if we have an HAO1 knockout individual who's an adult, does this inform their drug development? It turns out that the person we have with a knockout is a genuine knockout. It's in a 7 megabase autozygous region of the genome, so it's due to consanguinity that this rare variant, which is a frame shift and then a premature termination. And this lady turns out to be a healthy mother with 3 children. So we know the phenotype already. Here's the reads, and here's the Sanger sequencing, and we've confirmed that. And then we recall the individual for clinical history examination and biospecimens. And these are some of the measurements we've made, in particular the plasma glycolate. You'll remember HAO1 is called glycolate oxidase, and the plasma glycolate in this person is sky high. It's 12 times the upper limit of normal seen in other healthy individuals. The urinary glycolate creatinine ratio is also sky high. So actually this data alone confirms that the person is a genuine knockout for HAO1. We've done some cell line work. So if you transfect the variant into in vitro cell lines, there's no protein, there's no RNA, or very low protein. This is the wild type. You can see it forms in these granules around the outside of the cells. With the knockout, you don't see any expression, but if you do this at really high exposure, actually it's more diffusely localized in the outside of the cells, and so the protein, which is expressed at a very low level, actually mislocalizes within cells, and this is because the termination misses the signal peptide that causes specific localization in the peroxisomes. And then we did some metabolomics with Metabolon, who assayed 1,800, I think, metabolites and lipids within the plasma, and we compared this to 27 other individuals we'd recalled for the Genes and Health project, and compared how many standard deviations above the mean this individual's results were. And so the glycolate, which we'd already shown was high, is 170 standard deviations above the mean from the healthy controls. But there are a load of other biochemicals which are 5 standard deviations above the mean, so extreme outliers. And these include some other cholic acid compounds, which is a known function of glycolate, but also some other potential substrates of the HAO1 enzyme which were not previously known, like hydroxyisocaparate, which is greatly raised, but also its metabolite is reduced in this person, so suggesting novel HAO1 functions. So this is our first successful academic industry partnership. Alnylam are now moving on to phase 3 studies. We get a mention in their latest press release, and this individual with a knockout will go into their FDA submission should the trials proceed successfully. So finally, I want to just talk about genes and health. So this knockout individual was recalled under the Genes and Health project, and this is a project for long-term population genomic medicine in British South Asians, funded by the Wellcome with the Broad, Sanger, King's, Bradford, shortly Manchester. We've published a cohort profile in the International Journal of Epidemiology if you want to know more. But we set this up We're recruiting Bangladeshi and Pakistani adults from East London, Bradford and Manchester. We've recruited just under 50,000 people who give genomics, health records and consent for recall. And this is one of the reasons why we set it up. I work in Whitechapel in East London and I see the high rates of disease and poor health. This is using electronic health records from primary care. We have good access in East London from 1 million individuals who are GP registered adults, and just looking at rates of coronary artery disease. And if you break it down by age and ethnic group, you see particularly in relatively young Bangladeshi and British Pakistani people, the rates of coronary artery disease are very high compared to the white population, nearly 3 times in the 46 to 65 age group, which alas I now fall in. British Indians are a bit lower, and it's actually Pakistani men who have the very, very highest rates of heart disease in the UK. So this is why we set up the study. There are some other features, such as UK Biobank has recruited mostly white people, and so our study complements UK Biobank. We are on target to recruit 50,000 people by the end of this year and 100,000 people by end of 2022. There's a lot on our website, in particular the cohort profile. We've made some lists of phenotypes curated from health records, whether that's primary care or local secondary care. We've made a data dictionary, so you're welcome to look at these and see numbers of people with diabetes, for example, that we've recruited. In advance of this meeting, we put out a couple of days ago the latest list of knockouts, which is Hilary Martin's work, and this is available on the website. And this includes both the Genes and Health study in East London and now Bradford, but also the individuals from the Science 2016 paper from the Born in Bradford study and from Birmingham. And we have 2,300 genes with a rare homozygous knockout with the information publicly available. If you want the raw data, it's going to be on EBI-EGA. Much of it is on there already. We're just uploading further data. So that's exome sequencing data. We've exome sequenced about 5,000 samples from the Genes and Health study. I would love to do 100,000 whole genomes, please, on the Genes and Health study. If anyone would like to fund that, we have the samples prepared and waiting in UK Biocentre for you. At the moment, we're running Illumina chip genotyping on everybody, which actually we think will be higher quality than sequence data for heterozygote calls across the genome, and actually enable various other things like polygenic risk studies in minority ethnicities. So this is what's available. And then last slide, we're very keen, in fact Daniel MacArthur, who's one of our collaborators on the Wellcome Award, is very keen to make a human knockout browser, and we've been seeking funding for this. So this will, you can probably recognise this is quite similar to Nomad and uses some of the same software as Nomad, where you can take a gene and then look at the knockouts and look at things like age, populations, phenotypes, distribution and this will be publicly available. And we'd like to scale up. We probably won't find 10,000 genes with the Genes and Health resource alone, but if we combine Genes and Health with Promise, with Genomics England, with Saudi Genome, Qatar Genome Project, with everything else going on in a publicly available database, a bit like Nomad, we would get to all the human knockouts that were compatible with life. So that's one of our aspirations. I'll stop there.